to chapter 2. So we just finished talking about carbohydrates. Now we're going to talk about lipids. So lipids are the primary component of our cell membranes. They are hydrophobic, which means insoluble in water. That also means that they are nonpolar. So that's here, nonpolar, insoluble in water. These lipids are the most energy rich or dense foods that you can eat. They consist of carbon, hydrogen, and oxygen. So they are organic because they have carbon as the backbone. But again, remember they're nonpolar, so there's no charges, no difference between their head and their tail. That's what it means by nonpolar. It's insoluble in water. You cannot dissolve it in water. And we have three main types that we'll look at: simple, complex, and steroids. So this is a simple lipid. It's composed of a head, which is made of glycerol, and then three tails, which we call fatty acid tails or fatty acid chains. And because it's three, we would call this a triglyceride. You can have a monoglyceride, which would be just one chain on the glycerol. You can have a diglyceride, which would be two, and then of course this one in the picture is the triglyceride, which is most common in our diets. Now, the difference between saturated and unsaturated. So when it's saturated, it'll look like this tail here and this tail here, which are completely saturated with hydrogen all around. So there are no double bonds in this tail, all single. But this third tail has a double bond. And since it's only one double bond in this whole fatty acid chain, just one double bond means it's a mono unsaturated fat. If it had two or more double bonds along the chain, then it would be a polyunsaturated fat. Now, because of this double bond, you get this kink or bending in the tail, which now means it doesn't lay flat against the other two chains, and now it's taking up more space. This is why your oils like olive oil, vegetable oil are liquid at room temperature. But things that are saturated, like leftover fat from cooking meat, when it sits at room temperature, it becomes solid. So saturated fat is solid at room temperature because these fatty acid chains will stack flat and tightly against each other, and it'll be more dense. This saturated fat is considered like the unhealthy fat. Worst of everything is cholesterol, which is a completely different molecule we'll look at later. And typically saturated fats are high when you have animal fat, and then that's when you will also have cholesterol. Sometimes you can have some saturated fat from vegetable sources as well, but it would be in a much lower amount than an animal. And like I said, this is the monounsaturated fat. If it was poly, it would have more than one double bond. Last thing about this picture is that this is a natural form of the double bond where both of the hydrogen are on the same side. It's called cis configuration, same side. This is the natural way when you have a double bond in mono or polyunsaturated fats. They look like this, and that's how you get the property of being liquid at room temperature. Okay, but what we've seen is that we can have unnatural fats like trans fats where the double bond, I think I took that image out, but where the double bonds have hydrogens on the opposite sides, and then it would be trans configuration opposite, and then it will lay flat, but it's unnatural. Our body doesn't know how to process it. So that's what trans fat is. There's a lot more that we could talk about with trans fats. If you're interested, let me know. But for now, this is good enough. Okay, so for complex lipids, you're taking something like a glycerol and a phosphate, and then you're sticking on the fatty acid chains to that head. So this is the basic unit of our cell membranes. We call it a phosphate head, but it's actually phosphate and glycerol. And then you have your two chains of fatty acid. One of the chains is saturated, one of the chains is unsaturated. So complex lipids could have a different form for their head, and they could have 1, 2, or 3 chains, and they can be saturated or unsaturated. But the example that we're using for complex lipids is the phospholipid that you have in your cell membranes that forms the phospholipid bilayer of the membrane. Okay, but you could instead have mycolic acid, which we'll see later on in this class when we look at tuberculosis bacteria. Mycolic acids are a major and specific lipid component of mycobacterial cells like tuberculosis. It forms like an envelope, this like waxy coat around the cell. And that gives it extra protection from our immune system and from drying out. So it makes it hardier. Okay, so this is, again, phosphate head, fatty acid tails. One is unsaturated because you got that double bond. And this is what you find in every single cell in your body has a cell membrane made up of phospholipids in a bilayer. Animal cells have cholesterol in their cell membranes. That helps give it some extra support. Other types of cells besides animals will have, instead of cholesterol, they'll just have a sterol or even a steroid. So sterols are alcohols that are common in plasma membranes, and excuse me. Okay, I'm back. When you have a steroid, the steroid is going to have this basic form where you have 1, 2, 3, and 4 rings. Notice the first 3 rings are the same amount of sides, but then the 4th ring is only 5-sided instead of 6. And that's the basic structure of cholesterol. Cholesterol has a specific side group that makes it a cholesterol molecule. So this tells you that all of the steroid hormones in your body are derived from cholesterol. So even though we tell you that you don't want a diet high in cholesterol, you do need a little bit of cholesterol in your diet. Your body will synthesize the majority of the cholesterol, so that's why people can have a low cholesterol diet and still have high blood cholesterol levels, because their body is making more cholesterol than they actually need for their cells. And that's why you don't need to consume very much cholesterol at all in your diet. But anyways, all of the steroid hormones, including testosterone and estrogen, are derived from cholesterol. Furthermore, little amounts of cholesterol will be scattered throughout your cell membrane. And also, in case you've forgotten, the definition of a hormone is that they're a chemical messenger, right? They don't go inside of the cell. They land on the cell at a receptor, and that will activate the cell to do some process. And so sterols are kind of just this generic term for a cholesterol-like molecule that goes in the membrane. If you didn't have sterols to separate the fatty acid chains of the phospholipids, they would get packed a little too tight and they would become hard at lower temperatures. So adding the sterols into the membranes helps keep your membrane fluid instead of becoming hard. If your membrane becomes hard, then it's going to become like frail. It's going to be susceptible to being broken. Okay. I think that's all I wanted to mention. So sterols are an alcohol because they have the functional group hydroxy. Good. OH. The OH group. That's what makes it an alcohol, which you see right here. So cholesterol is a type of sterol, and there are other types as well, not shown here. All right, proteins. Every single cell needs proteins for the structure, the support, but also to carry out function. Most, if not all, of the function of the cell is due to proteins. Enzymes are a type of protein, and if you recall, enzymes speed up chemical reactions, and these are chemical reactions that would happen on their own but would take a lot of time and energy. Having an enzyme makes it happen super fast with much less energy. Enzymes are super important. Every single breaking down food and building together of all the parts of your cells, all the parts of you, all of that's done by enzymes. Okay. We have transporter proteins in our cell membranes that regulate and move molecules either inside or outside of the cell, right? So we could have things that are waste products we want to get rid of, or we have messenger molecules that we want to send out, and those can go through transporter proteins. Likewise, we have food and things that we need to bring inside the cell, and it can do that. Okay, however, when talking about proteins, sometimes proteins are bad, like bacterial proteins that act as toxins, for example, would be an example of a harmful protein. And again, recall that all proteins are made up of amino acids. Every single amino acid has that same basic form with the amine group. And then you can link hundreds of amino acids together. Once they are folded into a three-dimensional molecule, then they can be a functional protein. So amino acids are the building block of proteins. And this is what I mentioned earlier. You have your carboxyl group and you have your amino group, and that's what makes an amino acid. The R group will be different for each actual amino acid. So tryptophan and lysine, right? They all look exactly like this, and they only differ by this functional group where the R is. Okay. We also have right and left what we call isomers, and it's the L isomers, always L isomers in proteins, with the exception of glycine. Glycine doesn't have a right hand, it doesn't have an isomer, but everything else, all the other amino acids, 20 of them, are going, the one that you use in your body is going to be L. Okay, this is really important to take into consideration when you're doing any type of drugs manufacturing drugs, you always need to remember isomers, which isomer is actually used in the body so that you get enough of the actual usable form of the drug. Right, so there are 20 amino acids. You do not need to memorize them, but you do need to memorize this generalized form of the amino acid, and that it's the L isomers that we use. Okay, all of the amino acids differ by their side chain, which goes in the R spot. Okay, so a D amino acid does occasionally occur in nature, like in certain bacterial cell walls or certain antibiotics, but for the most part, we only care about L isomers. Okay, so I keep telling you that the amino acids differ by these functional groups, and so here you can see what that looks like. I also told you that depending on the functional group, it's going to give different types of properties to the amine group. So we see here amino acids with electrically charged side chains. So these are positively charged, these are negatively charged. Those are going to interact with their surroundings very particular way, right? Whereas these are uncharged side chains, and these are special cases, special properties, like having sulfur. Okay, and then here finally you have a hydrophobic side chains, so these would not be interacting with water, and so this makes a big difference depending on which amino acids are in a chain, determines how it folds and how it functions in the cell. Okay, so again, don't memorize all 20 amino acids, but just want you to get the overall concept. So a peptide bond is what links each amino acid to the next, but as I promised you, they're formed by dehydration synthesis. Okay, so here's amine group, here's a carboxyl group, and every time you see an R, that's a functional group that's not being shown. So you have 1 amino acid, 2 amino acid, 3 amino acid, 4, 5 amino acid, 6. So this is a chain of 6 amino acids. Every red spot is a peptide bond. So the peptide bond forms between the carboxyl group of 1 amino acid and the amine group of the next amino acid. So you see a carbon bound to a nitrogen for every peptide bond. Okay. Furthermore, this looks flat, right? There's no folding yet. It's just linking the amino acids together. So this is called the primary structure. It's just the amino acid sequence. Then you get a secondary structure when you start to get a little bending, a little bit of folding. You might get a helix or what we call a pleated sheet, and that's a secondary structure. This is still not a functional protein, but we're getting closer, okay? Now we have tertiary structure when those helix and pleated sheets get folded together, and you get a 3D shape overall. And it's not shown very well in this image, but what they're trying to show is that you'll get a lot of different types of bonds happening in this level. You can get, you have regular hydrogen bonds here. You have a hydrophobic interaction happening here between the side groups of different amino acids. Here you get that super strong disulfide bridge that I was mentioning earlier. And then here you have, a positive and a negative charge interacting. So this, where it says disulfide bridge between cysteine molecules, that actually should be up here. I think going from maybe an older version to a newer version of PowerPoint, it kind of got displaced. So make a note that this disulfide bridge is actually here, where it says S. Okay. So tertiary structure includes all the types of bonds: hydrogen, ionic, disulfide, everything. Okay. You can read a little bit more about each level of protein structure in your book. Quaternary, most of our proteins will have quaternary structure, though it's not exactly required for function, meaning that we do have some proteins that only have tertiary structure that are functional. Most of them will be quaternary, however. So that's 2 or more folded polypeptide chains get put together. And furthermore, we also tend to modify our proteins. And so this is where this slide comes in, other proteins, conjugated proteins. So you can add sugars and maybe even a nucleic acid, lipids, and phosphates. So all other types of molecules can be added onto proteins to make them more complex and have more function. Okay, hemoglobin is an example of a conjugated protein with the heme group. Phosphoproteins may be important for the survival of bacteria, such as Legionella, that grow inside of a host cell. There's lots and lots of examples of proteins with one or more of these additional compounds added to them, and you'll see a chart of that in your book as well with some examples. Okay, so I asked you, what effect do you think heat has on a protein, such as an enzyme? And what hopefully you would say is some heat will speed up action, but too much heat will start to break those hydrogen bonds, possibly other bonds, and then you will lose the shape. And if you lose shape, you lose function. So a little bit of heat can be good, can speed up molecules, move faster, so you get more reactions. But too much heat will, what we say, denature the protein. What does it mean to denature? It means that one or more bonds is being broken, and that you get a loss of shape. And remember, if you don't have that perfect shape, you're going to lose the function. All right. So I think these slides got mixed. I skipped over this and went to this slide, and then I came back to it. So if you're looking at your slides and you're a little confused, it's just a simple little switcheroo there. Okay, another question for you. What 2 functional groups are in all amino acids? Carboxyl and amine. Okay, so nucleic acids, you're probably pretty familiar with, again, from all of your previous coursework. So DNA and RNA are both nucleic acids. They are polymers of nucleotides, right? You have hundreds and hundreds and hundreds of thousands of nucleotides that make up the sequence of DNA. And when we talk about DNA, we tend to talk about the nitrogenous bases in particular, A, G, C, T, or U. You only see U if we're talking about RNA, and you will see T if we're talking about DNA. Okay? All nucleotides consist not only of the nitrogenous base, but they'll each have a sugar. It'll either be a ribose sugar if it's RNA, or it'll be a deoxyribose sugar if it's DNA. Each nucleotide attached to that sugar is going to have a phosphate group, and this is why we call it the sugar phosphate backbone of DNA. And then the nitrogen group will be either AGCT or AGCU. The A and the G's are called purines. CT and U are called pyrimidines. As I mentioned, DNA has the sugar called deoxyribose, and deoxyribose has one less oxygen. D, less, oxy, oxygen, ribose, sugar. If you look at RNA, just skip ahead for a second. If you look at the RNA sugar, which we see right here, it's a ribose sugar, and it has the oxygen here on the second carbon, okay? So let's go back to DNA real quick. You start from the oxygen, you go to the right, you count 1 and 2 carbons. You're missing an oxygen, that's why it's deoxyribose. But notice 1, 2, 3, 4, and the 5th carbon's hanging out above on top. Okay, that's real important for learning the structure of DNA. And then you'll notice that on that 5th carbon that's hanging out on top is where the phosphate group is attached. On the 1st carbon is where your nitrogenous base is attached. Okay, all right. This is the double helix. It's 2 strands of DNA. What are all these dotted lines? Those are the hydrogen bonds. You'll notice, not in this image, maybe, yeah, I don't have an image, but in your book you'll see an image where I'll show you that you'll have more hydrogen bonds between C and G and fewer between A and T, and that's how you get this, that and the different lengths, different sizes of the nitrogenous bases, all that contributes to the exact shape or this irregular shape of the double helix. Hydrogen bonds, as I've said over and over again, are what hold the double helix together. The one strand is held together to the second strand only by these hydrogen bonds. So we've talked about hydrogen bonds require heat to break. So this means that if you add enough heat to a solution with DNA in it, you can cause those strands to come apart. And that's an important concept that will come back up when we talk about PCR. All right, so RNA, how is it different from DNA? Well, it's single-stranded. Okay, so that's important. It has a U instead of a T. The sugar, we already talked about the difference. On the second carbon has the OH instead of just H. We copy a gene of DNA into RNA, which we call messenger RNA, and then that is used to make the protein. There are other types and functions of RNA, but the majority in introductory level courses will be focused on messenger RNA. RNA is also considered less stable than DNA. So whenever you're working in the lab, you will want to work with DNA instead of RNA whenever possible. Okay, lastly, ATP. ATP is cellular energy. So you break down your food, every time you break a bond in a food molecule, you release some energy, and at the end of the entire process, which we'll look at in a few chapters when we look at cell metabolism, but in the end, you're going to get enough energy from breaking these food molecules to force a third phosphate group onto this molecule to make ATP. Phosphates are what we call highly electronegative. Look at all this oxygen. They don't like to be together, and especially 3 of them together. Okay, so this actually holds a lot of energy. A lot of energy will be released when you break this bond right here to free off one of these phosphate groups. So that's cellular energy. It's safer for us to work with energy in this form than in the form that like our food comes in. And so whenever you store up energy, you store it up as glycogen, sugar, or fat. Then we can quickly change that sugar or fat into ATP. We never store up ATP. It has to be used pretty much as soon as it's made, it's going to get used, okay? And the energy, again, comes from breaking off that third phosphate group. Nucleic acids and ATP look pretty similar, right? So adenine, you saw over here in this nitrogenous base could be A, C, G, or T, and the A stands for adenine. Same thing happening with ATP. It's adenine, but when we're talking about ATP, we call it adenosine triphosphate because it's attached to a ribose, which is attached to 3 phosphate groups. So this is ATP, but it looks very similar, has the same kind of building blocks as a nucleic acid. Okay, so phosphate connected to a sugar, right? Phosphates connected to the ribose sugar, and then the specific nitrogen base is adenine when it's ATP. All right, and it's 3 phosphate, triphosphate, 3 phosphates. So that's it for ATP. Here's some check your understanding questions, right? What does DNA stand for? What does RNA stand for? How do DNA and RNA differ from each other? What are the components of a nucleotide? What do we mean by complementary base pairing? I didn't actually talk about that, so let's go ahead and look at that real quick. Every time you have a C, you will see it pairing with a G and nothing else, right? Every time you have an A, it's going to pair with a T. So that's complementary base pairing. That's how they bind. The hydrogen bonds form between A and T, and they form between C and G. You cannot mix the letters up, right? A goes with T, C goes with G. All right. So would you be able to tell me the second strand if I give you a short sequence of nitrogenous bases? Would you be able to tell me the complementary strand? What are the 4 nitrogenous bases and how do they bond together to form a double helix? And how does ATP differ from a nucleic acid? All right. Good job. You are now ready to review your notes, read the chapter if you haven't read it yet, and do the practice questions at the end of the chapter. Have a good day. Bye.